Right, so yes, I'm Andrew. I'm from SFU. My research project concerns emerging diseases of intensive organic greenhouse vegetable production. And today I'll be talking about this post-harvest infection, mostly a black spotting of tomatoes that's become a big issue in British Columbia. So first I'll talk a little bit about the greenhouse environment and the type of media that we use commercially in organic greenhouses, because there's a few nuances that could be contributing to the disease that I'll be talking about. I'll outline the disease that we're looking at, tell you the objectives of the current study. I will tell you about some of the pathogenicity testing that we did using our what we think is the pathogen. We also looked at just generally at tomato surface microflora in organic greenhouses. And then I'll give you the conclusions and tell you what sort of future directions we're looking at for this line of research. So to begin with, the organic greenhouse environment, so just a bit of detail on these greenhouses that I'm talking about. I work with about 50 acres of certified organic tomato production. That's intensively managed and has been ongoing for several years. When I say it's fungicide-free, obviously we still use inorganic fungicides like copper and sulfur derivatives. We just don't use synthetic chemical fungicides, which then of course are organic chemicals, so it leads to a confusing homonym. And then we use biofungicides as well. The tomatoes over the course of the growing season will reach tens of meters tall, so workers are constantly de-leafing plant matter onto the greenhouse floor, and so this leads to the accumulation of a lot of biomass on the greenhouse floor. And then the soil media, it's sort of like the soil media we've seen so far today. It's intensively managed, it's very nutrient rich, and so the plants grow at a very high speed. This is what the media looks like throughout the tens of acres that these tomatoes are grown in. This system sometimes is containerized, like the ones that you've already seen. A newer system that we use now, this is still fertigated, and the recirculated nutrient feed can be treated and reused. But all the media is laid out flat on the greenhouse floor, and it just makes everything easier operationally. But the important thing here is that this media is very biologically active. You can see the top of the media is completely colonized by actinomycetes and fungi. And so I'm putting this out because the moving back into soil media in the greenhouse is not a simple process. It was the soil-borne pathogens that precipitated the move to hydroponics in the first place. Things like fusarium, but also Parenicida causing corky root rot. So now going back into the media, we have a whole bunch of other concerns. In addition, the more difficult biochemistry of the soil and all the microbial activity. So the greenhouse media, the soil media, is usually what's blamed for any increased disease incidence in organic greenhouses versus conventional greenhouses. Just to quickly show you what the leaf litter looks like, this is not that much leaf litter for the greenhouse. This is actually a conventional greenhouse. You can see that where the soil media is in the organic greenhouse, that's where conventional growers put the leaf litter so people don't walk on it and aerosolize all the spores that are sporulating off the litter. You can't do that in organic production because that's where the soil is, and it's very difficult to use the kind of raised system that conventional growers use. So it's kind of a small operations detail, but then these little nuances do have huge ramifications for disease, even if they're just small management decisions like this. So, in 2009, we saw a black spotting on tomatoes in BC, and it started to occur at a very high incidence and cause very large crop losses because it would happen within 5 days. The tomatoes would be so heavily infected that they couldn't be sold. They would just be shipped back to the growers to be composted. This is confounded because the organic product is sold as a premium product, and so consumers don't want to see, at least for this particular product, don't want to see blemishes on the product. So when this disease is a blemish, that becomes an issue. So I'll just quickly go through what the disease progress looks like. So we start with this very small, minute black spot on the tomato, less than 2 millimeters. We can start to see the lesion become a little bit sunken. Then the lesion will develop a yellow center. The lesion will progress to become larger than 3 millimeters, still with the yellow center, and the lesions might start to coalesce. Oops. And here you can see a tomato that's pretty heavily infected. There's lots of lesions that are coalescing with these large yellow centers. And then we see in the middle of the yellow centers, a little bit of mycelial growth starts to occur. And then eventually there's secondary infection and more mycelial growth on the surface of the tomato. And then you can get things like botrytis, nesting, and rhizopus and other post-harvest diseases that usually occur at low incidence in the greenhouse, but become more devastating. And then even sometimes arthropods will get involved and make the situation a lot worse by vomiting on the tomatoes, and it just gets really gross. So our objectives were, first, we wanted to identify the causal agent of the disease. Then we wanted to investigate the effect of temperature on disease severity. We wanted to look at inoculation position and the ripening stage of the tomato on the disease severity, and also the effect of wounding on the disease. We also wanted to survey the tomato surface microbes in the organic greenhouse environment. And then we wanted to investigate the pathogenicity of all of these other ubiquitous microorganisms that we found. So we found lots of tomatoes with these lesions. So we would excise the lesions, surface sterilize the tissue, and then we'd plate that out onto non-specific media. And predominantly we get, like you can see here, lots of different types of penicillium. And we were able to, based on the conidiaphore morphology and the colony morphology, separate the penicillium into 3 basic groups. The first one was a gray one, and then there were 2 other ones, a green one and a more bluish one. So then we used PCR to identify them to species. Penicillium are difficult to identify to species, so we used the internal transcribed spacer and the beta tubulin, and sometimes we had to use the cytochrome oxidase genes, so we'd sequence those to figure out what it was. It turned out the most common one was Penicillium olsonii, and then we also found Penicillium solidum and Penicillium polonicum. So we would take tomatoes, we would inoculate them with drops of a standardized conidial suspension, and then we'd incubate them in human chambers for 7 days. We'd calculate a disease severity index based on where 0 would be no lesion, 1 is the tiny lesion that I talked about, 2 would be kind of an expanding black lesion, and then 3 would be the severe yellow coalescing lesion. And what we found with Penicillium olsonii was it was actually reproducing the same disease that we were seeing in the greenhouse and in the packing houses. And it was more severe at cold temperatures. So cold temperatures in this case are 12 degrees Celsius. With the other Penicillium isolates that we had, they were also worse in the cold. But these ones would tend to cause rapid decay of the entire tomato and soft rots. So these were not like the symptoms that we were interested in. They were far more severe. Penicillium polonicum, also pretty gross what it would do to the tomato. So these ones, we weren't as concerned with them, but they occur in the greenhouse and they probably are causing these diseases, but at a very low incidence, so these tomatoes are just thrown out. Whereas with the Penicillium olsonii black spot, pallets and pallets and pallets of tomatoes were coming back with this mild disease, but at very high incidence. So when we looked at whether the disease was worse when we wounded the tomatoes, we would just take a sterile insect mounting pin and wound the tomato before we'd put the conidial suspension onto the tomato. And you can see, as you'd probably expect, if you wound the tomato, sometimes the disease severity is much higher. Obviously, it's going to let the pathogen get through the thick tomato cuticle. The interesting thing here was that it didn't increase the disease severity in every case. So the wounding is not necessary for disease progress. And we thought at the beginning, maybe there was some insect feeding happening, wounding the tomatoes beforehand, but it looks like that's not the case. This shows two different effects. First of all, the ripening stage. If we were to infect the tomatoes at the breaker or pink stage, disease severity would be much less than if we inoculate them at the ripe stage. And at the ripe stage, where you can see disease occurring, the disease is worse on the tomato shoulder than it is at the equator of the tomato, and these tomatoes would have also been inoculated at the base. You can see very little disease anywhere except on the tomato shoulder, which is where it happens in the greenhouse, and is often the site of russeting on the top of the tomato, and it's also where the calyx touches the tomato. When we section and fix and mount and take microtome sections of the tomato lesions themselves, we found they were often centered on microcracks in the tomato surface on the shoulder. So you can see at the top, there's a bit of a microcrack on the tomato surface, but that one's uninfected, and then on the bottom, that one is infected. So you can see the necrotic tissue underneath. And so the black spots were usually centered on these microcracks in the tomato surface. We were also interested to see what organisms were present on the calyx. We thought that might be playing an effect on the disease progress. And indeed, the tomato calyx would often have the same penicillium populations that we were seeing coming out of the wounds. When we looked at the general tomato surface microflora, sometimes we'd have plates with isolated colonies that we could count. But you can see all of the green colonies on these plates are all penicillium. And the way we got these is we would go in the greenhouse with sterile swabs, swab the surface of the tomato, and then just immediately plate it onto potato dextrose agar, and then incubate it for 7 days and count the colonies. Here you can also see the yellow colonies are aspergillus colonies, and the black ones are probably cladosporium or aspergillus or some other dark phyloplane fungi. So we would find Penicillium wilsonii, Penicillium solidum, Penicillium polonicum, just like when we isolate it from the lesions. Rhizopus, a known post-harvest pathogen. Alternaria, aspergillus, cladosporium, geotrichum, probably all causing disease at low incidence. But those ones, when they do occur, are more severe, so those tomatoes are probably thrown out before going to market. You can just see the populations of the various tomato surface microflora would vary from week to week, but consistently we would get these groups occurring in large amounts on the tomato surface consistently week after week. When we did a little bit of pathogenicity testing on the other microbes that we found, these ones were actually helped by wounding, whereas Penicillium wilsonii generally wasn't. So that was an interesting result. Is that it? Yeah. So we concluded that black spot development caused by Penicillium wilsonii is influenced by temperature, by inoculation position, and by the ripening stage of the tomato. Wounding usually increased the severity of the disease for all the other organisms that we isolated, but not for Penicillium wilsonii necessarily. So we conclude that Penicillium wilsonii is a weak pathogen. It takes advantage of the natural russeting on the tomato fruit shoulder, and along with other post-harvest spoilage organisms, it is present at high concentrations in the organic greenhouse. So we want to figure out where the post-harvest spoilage fungi originate in the organic greenhouses, whether there's a strong effect with the leaf litter, whether there's a strong effect with the soil media. With this, we should be able to develop expert management strategies for controlling this disease in the same way that the growers use management strategies to control botrytis by timing all of the worker activities based on humidity in the greenhouse and other effects. And then we would also like to look for control measures for this type of post-harvest spoilage, whether those are things like biological controls that might help control the penicillium populations on the tomato fruit surface, or whether they're things like physical treatments that can induce a defense response in the tomato, and that could be something like UV irradiation or something else that would be acceptable for organic production. All right, so I'd like to acknowledge NSERC. I have an IPS along with Harrio Research, which is a company in Langley, BC, BC Greenhouse Growers Association, Growing Forward, Origin Organic Farms, and Village Farms for supplying tomatoes and letting us in their greenhouses. Yeah, and Anna, who helped with the project. Thanks.